with this, I would like to actually thank Ms. Ho Yuat Me very much for inviting me and giving me the privilege this evening. So what I will do is, first thing first is I'll share my screen, and then we shall begin. Okay. Okay. I think the screen should be okay by now. Yeah. Okay. So the reason why I chose this title is because we have gone through a few what do you call this geologic periods. So it started off with the 4 billion years ago when Earth was made, and then we divided it by geologic timescales. And within even before the Jurassic, even before the Triassic, came the Quaternary and came the Ordovician, and then right now we are in Quaternary. So within that time frame, it's around 250 million years to 450 million years. So that is where we start seeing the horseshoe crabs emerging. So with that, I will start on with the introduction of my team. I've been working with these marvelous people for a very, very, very long time, ever since I started this passion with this animal. And along the process, we also got a few members coming in, and there was some empowering, two-way empowering ourselves and as well as them. And with that, we are now having a very, very strong team working with us. And along with that, I would like to also introduce the animal actually, aside from the team, now the animal. We have this organism which is known as an arthropod. This arthropod is a very unique arthropod because it is not closely related to the crab. It is not closely related to the shrimp. It is falling under the same category as all the other mammals with legs, and then it is further divided. Okay, so under this, it is on its own class known as Merostomata, and from this group we have a branch out of scorpions, spiders, ticks, and that's where the horseshoe crab actually is situated. So why they put the horseshoe crab there? It's simply because being a Merostomata, it has, we can call it 6 pairs of legs, 2 that are available at the mouth, and then 1 is modified. Usually for the male, it is for mating. For the female, it is for searching for suitable grounds to penetrate in and burrow. So I will go on with that. And then the next thing is the exoskeleton. Of all the organisms that we know, this organism, it is non-calcareous. It is made of chitin and chitosan. So it's a very thin layer of material, but it is binded by 2 different materials, something like what the squids have. So squids are also very ancient. This organism is very ancient. So this primitive texture is available on them. And likewise, when you look on the spiders, you look on the scorpions, they are also having the same trait. Okay? Then here is where Merostomata plays a big role. It is it divides. This term is given to an organism that is divided into 3 sections. So we have the carapace, we have the opistosoma, and we have the talson. So the talson is always the tail. The opistosoma is where the gills are. So in this image, you can see the gills. And then we have the carapace where the appendages are. So based on this classification, the organism is now taxonomically derived and placed together with spiders, scorpions, ticks, just for that reason. And the most important thing through the 11 years experience is the image on the left. Everyone was dividing the horseshoe crab by their species based on the tail structure. Okay, but When it came to the gender, they only focused on the first modified appendage. And that itself was a big no-no when you're classifying larvae stages, because larvae stages, they appear very identical. So for that, the horseshoe crab is now divided using different keys so that we can differentiate them when they are young, when they are old, and also if there's any form of damage. So with that, the 11 years of experience is the image below, where we started looking at the genital flap. Okay? So when we flip over the first flap, that is the genital flap, we are able to see the structures. If it's rounded, it is a female. If it's sharp, it's a male. Then the next course of experience comes in differentiating each type of crab that's available in the area. So when we communicate with the fishermen, they will term the word blancas for all the horseshoe crabs. But right now, because of our constant engagement with them, they're able to differentiate the round tail and the triangle tail. So when you see Onychacinococcus protonicauda has the rounded tail. The others like Limulus polyphemus, which is available in America, and it's the only American horseshoe crab. Okay, it's the only exclusive species. Asia has 3. It is divided into Cassinoscopus and 2 types of Tachypius. One is the Gygas and one is Tridentatus. So all of these, which are non-Cassinoscopus, their tails are triangular, and the Rotundicaуда is the only one with a rounded tail. And then when we look on to other morphometrics, what we learn is that their genital operculum also appears differently. Their carapace front, when we put them facing us, it also appears differently. And the spines, the larger they are, the longer the spines appear. So based on this experience, this was where I share with you a picture. This is how I teach people who want to learn about horseshoe crab. We do not rely entirely on the appendages. Supposingly they got damaged or there's net capture, then that will be a big problem. So I will be starting off with this. When we have a female crab, of course, size plays a big role. Females always appear larger than the male. And when we flip over the genital operculum, we just want to confirm. And there itself is the first form of engagement, where we use this word touch and feel. The moment they touch the crab, they engage themselves into curiosity, and that's where the learning process begins. So for this session, the learning session, although we cannot touch and feel, but I can give you all a visual impression on what you all will be looking at the crab. So when you flip over the genital operculum, the moment you see this structure, it's rounded, it's a female. The next thing what you all will notice is the eggs. If the eggs are available at the genital operculum, like in this image, the crab is mature, it's an adult crab, it's pregnant with eggs, and it is ready to mate. So how do we differentiate whether this crab has mated or not? We will flip over the crab and we will look for mating scars on the opistosoma. If there are blackish signs on them or some sort of damage on them, which appears in a black coloration, it means that they have already experienced their mating. But if they do not have that, they are known as newbies. So newly reaching the adulthood. So the next thing when we get curious about the crab is to differentiate the crab. Now, this came with experience. How do we know whether the horseshoe crab is also actively participating in reproduction? For example, taking this image, we notice the first thing is there's no mating scar, okay, because of lighting and all those errors. But If you look at the visual structure, this is what a mating scar will look like. Okay, it's a black damage. But over the years, we got a little clever. What we did was we started looking at the spines. If these spines are proper, very healthy, and it's a complete set of 6, it means the horseshoe crab is not mated. This is only for female, not for male. For male, you will get the full 6 set. But for the female, this will be very short. So when, let's say, you want to get to know the horseshoe crab, go to a restaurant or go to a wet market, and you will be able to see the crab. And then from there, you pick up the female, and then you start examining all these kind of structures. Okay, so that itself will tell you roughly how old is the crab. So the horseshoe crab takes about 14 years to mature, maximum, and is active for reproduction for about a span of 19 years. So in that means an additional 5 years, 19, you will start observing encrusting on the organisms. The moment we get fouling or encrusting, means the crab is already old and no longer fit for reproduction. So that's another trait that we can pick out when we are looking at crabs available for sale. And this key is very important, especially when you do bycatch assessments, because if you're not having encrusting, it means that the crabs were caught in the deep sea. If they are having encrusting, it means the fishermen are catching fish, catching other crabs, and this came along. Okay, so they appear solitary and slightly weakened. So now, knowing about the situation of the crab, we also need to divide the crab into a few sections. And according to a few years of experience working with the Asian species, the most important for the female will be the interocular distance. So the interorbit length, the distance between the interoculars. So that orbital length is only useful for measuring the female crab to say whether they're growing in the right size or not. Whereas for the male, it will be the proximal width. So the wider the width is, it means that the crab is experiencing a rough environment. So it needs to have a larger surface area in order for it to sink. If it's too narrow, it will always remain afloat and not able to mate. So these are the few keys that we are picking out now. And recently I'm working on solving this puzzle using statistics. How powerful can we use this kind of statistics to say that this population is going to be viable for a very long time? And that is the problem actually now we are facing, because we are removing too much of female crabs. So the males are going around solitary, unable to copulate, and therefore they remain solo. They have to feed, they have to endure the little bit of stress, and because of that, they appear a little bit weaker. Okay. So now, inside the crab, when you break up the crab, you won't always get eggs. So unless it's a female, and that also a female that is ready to reproduce. Otherwise, the eggs are absorbed internally, and it will appear as a yellow color sponge. If the male, it will appear as a white color sponge. Okay? So what we can notice is these few structures that are available when we slaughter the crab, and these are what scientists are using to understand their growth, their feeding habits, how healthy can the horseshoe crab get, okay? How favorable is the environment? These are the questions that they constantly ask themselves. Okay, and the next thing that I would like to reveal is when you slaughter a horseshoe crab, you will notice this blue color substance. This blue color substance is not in liquid form. It will appear like an agar-agar. It's how to say, an agar-agar that doesn't harden well. It needs a little bit of cooling. So that kind of agar-agar is what we call coagulation. The horseshoe crab blood, the moment it contacts with the air, it starts coagulating, and this is what we will get. And why it has this blue color, it's because of hemocyanin. It's a blood pigment. So this pigment is very exclusive to some organisms, but for the horseshoe crab, it's appearing as blue. So other than that, um, scientists, the image here is quite an old image from the 1800s. So they drew the horseshoe crab elementary, and then they started giving it color just to differentiate how does the internal structure look like. But when we run a histology sectioning, we can actually piece out together this information nowadays. But at that time, they did not have that. So they divided the horseshoe crab into few sections, and then they started drawing. Okay, so this is the early form of imprint on how does the organism look like inside. And then the modern ones will have the digitalization. And for those that are curious to know how does the horseshoe crab look like, go to restaurants, go to fishmongers. When they are chopping up the horseshoe crab, that is where we are able to see some structures. Now, why are horseshoe crabs not classified with other crabs? So we have crabs that are known as true crabs, and then we have like Malacostraca, those are true crabs. Uh, Over here, we will on the right side, we'll have crabs which are classified as the non-true crabs. The reason the differentiation between the crabs, I'm not expert on that, but what they said is based on the function of the pincers and the appearance of the body. So this is how they did their classification. But to us, when we look at crabs, we just classify them as crabs. Taxonomy is no better. So now the question is, why are they not together with all the other crabs? It is simply because earlier I've mentioned about the two-layer structure on your body, the chitin and chitosan. So when they mold, the mold skins are what you see on the left. Okay, and recently they did some work on how this structure should look like, and they noticed that it's like sponge-like, and these structures are not available on crabs. For crabs, it will be made from calcium oxide. That's why it's hard. But for horseshoe crab, it is very soft. It is flexible. It is porous. There's like air bubbles within it, but it's I wouldn't call it actually like air bubbles, but it looks like a foam. Okay? Then it's flexible, it's smooth, and it's fragile. You can actually break, you can snap the horseshoe crab carapace. Okay, you don't need too much of energy, but for the crab, yes, we need some strength. So this is how we differentiate horseshoe crabs from the crabs. And now we get on to the topic. How did we get from Ordovician to Quaternary? We were looking at structures. Okay? Along with that, we were looking at shoes given to horses. And then we said, okay, this looks about uh, like a horseshoe. So we gave it the name horseshoe. And then why did we call it a crab? It's because of the pincers. It has pincers. Okay, so simply we gave it a common name. We can't be calling it something else because the organism is not pretty. So we had to classify them with something that is not pretty, which is like the crabs. So based on the shape, the appearances, fossil records, and that was where the horseshoe crab was derived as a horseshoe crab and not as a trilobite. But if you look at the fossils, we are looking at trilobites. They are broken up into sections. They are very primitive. They have a single elementary system. They have a single central nervous system, and as well as a pulmonary system. So central nervous system is the nerve. Pulmonary is for the heart and the what they call is the gills and etc. So with these primitive kind of structures, uh, the horseshoe crab stands out on its own. Even in the fossil records, they have their own classification. But for the modern horseshoe crabs like Tachypus gigas, for the limulus, for the Cassinoscopus, these are the modern names that we are giving it to them. They are classified as the living fossils. Okay? They may look similar to their relative, but their relative has a different name. Then we move on to why they are known as living fossils. Because they appear similar, they take on the same form. The size is roughly similar. Okay, the only difference is they don't live forever. They have a lifespan. So we are not accurately able to define how long can they live. But based on hatchery experiments, keeping them in a confinement over a certain time, they have come up to say that the limit is 19 to 21 years. It cannot go more than that. So the next thing that we need to know is, other than the age and the appearance of the horseshoe crab, we also need to know what goes on. How do we actually encounter them? So we have to go to the beach. It has to be a lunar day with a high lunar tide. And with that, uh, if we go in the evening, um, high chance we're able to see something like this. It's a very unique display. I don't mind showing a video, but I'm only scared in my leg. So I've extracted what you will see on the video. So first is you will see a very happy couple coming to shallow water, and then they will be just looking around. And once they find a nice area, they will start digging. And when they start digging, that's where you'll start seeing bubbles coming up. Okay, bubbles will be coming to the surface like a scuba diver. A scuba diver around 3 meters or 2 meters, you can see the air bubbles coming up. So that's what you'll see. And when they start digging, then the bubbles become vigorous. Okay? Then when they have reached a certain depth, they stop that and they slow down. So you can see very small microscopic bubbles coming up. And then when there's no bubbles, that's where both the couples are laying their eggs. So this entire display takes about 2 to 3 minutes. And if you wait there for how long the female burrows and removes the egg, it sometimes can take up about 10 minutes. So we will just wait there. When the crab moves away, we will mark that area so that during the low tide, we can go and investigate how many eggs were removed by a female crab. So when we count the number of eggs and number of nests per female crab, then we will use the word fecundity for the female. So fecundity studies are also another separate approach that people use to find out and see whether a population is able to be sustaining for a long time or not. The more number of eggs, the more number of nests per female crab is a good sign. But more number of nests with very less number of eggs is a problem, because it means the environment is less favorable. The grow out rate is very poor, and therefore the female doesn't take the risk of deploying many eggs in one burrow. Okay? So the next step is we look at the general cycle. When you go to a mangrove area, you'll be getting all sorts of biodiversity. And then from there, we will be picking on certain spots where we think it can be a juvenile spot, a nursery ground, and we are able to get the small crabs, okay, small horseshoe crabs. This is Bailaka. So far, my only successful is out of Malaysia. In Malaysia, my students are successful, but not me. So hands-on experience for me, no. But when it comes to finding horseshoe crab spawning grounds, that's where the entire team works together. We map out roughly which area has sand patches, how does the sediment texture go about, how deep is our footprint. That is a very good key that we use to find out which areas are suitable spawning grounds. So with that, I've adopted this picture. It's published from an article. So they have differentiated how does the environment look like. And this good image is an overall, it's like an overview. And therefore I've included for this presentation so that it's easier to understand. Okay? But in my case, this is what I see. I can only tell if the imprint is depressed or this way. If it's depressed, it means that it's a new nest. It's roughly less than 3 days because we follow the lunar. We will go on the night of the full moon. We will go on the following day, which is a full moon day. And then it goes on to become like a semi-circle like that. So that entire span roughly is about 3 days. So this is where we come up with the numbers of 3 days. This is what we will see. If we come late, This is what we will get. We will get a bulge. Now, how does this bulge form? Because water keeps coming to the higher shore and again residing to the lower shore, the horseshoe crab eggs start separating themselves within the nest. Okay, initially they are one clutch, then they start separating. When they start separating, there is need for filling up wherever that is porous. So wherever that is filled, the sediment cannot sink back, and therefore it remains on the surface like a bulge. So this is what we will get for nests that are very old. So let's see what's inside the nest. So for clutches, this is what you get. You get everything clumped up together. And then what our job is, we will go to this clutch, not touching them with our hands, but with the spatula. And we will be counting how many clutches are there. Okay. The luck is not so good because sometimes when we remove a clutch from the sediment, we may separate eggs in that process. It's easier to separate and it's not easy to put them back in the clutch because once they get removed from the clutch, they cannot be put back. Okay? So this is what roughly we go through the entire process. But what we know is when we break a clutch into two, we can piece it back together, but we cannot stick it into one clutch. It will still remain as two clutches. So that's where we will count it as one clutch. So we divide it, we put it on the sieve, and then we start counting how many number of eggs are available. Okay. So to reach there, we will have to put it into a 2 mm sieve, but that depends on the grain size that's available in the area. Some areas we will need a 4 mm sieve, some areas a 2 mm sieve. So we will put these eggs over there and then we will vigorously move it left to right. Not too rough, just nice so that we get some bubbles. Okay? So when we have this kind of bubbles, it means that the sediment is being sieved out. The bubbles are formed by organic materials. So nothing to worry when you do that. It's a common practice. But after sieving, on the right image is what we will get. So we will use toothpicks and move the eggs aside as counted, or if they are appearing as larvae, we will follow from top to bottom order. So this methodology has been practiced by our team for a very long time. And so far we are successful in getting the counts right. But if it's too many number of nests in one area, the work can go on for hours. Sometimes during the low tide and we are getting the tide really coming near to us. So it can go through that as well. So doing this is actually, we need more manpower. A team of 4 people is just nice. A team of 2 people, it can take very long. So these are among the examples that we do when handling the horseshoe crab. We try not to contact with our fingers. Now the next slide is where I will tell you all why. So this was during my early training. Okay? So without any set of skills, we just acquired the horseshoe crab with a spatula. After sieving, we put it back on the spatula, and sometimes the eggs get into contact with our fingers. So putting it into the holding tank, bring it back to the laboratory, and this is what happens. From eggs that are very clean, they start becoming sticky to sediment. Okay, so they become sensitive. And then keeping them for some time, you start getting fungus, okay, on day 9. So we will have to remove the eggs that are having fungus. So it means, like example, from a set of 100 eggs, the moment they start getting fungus, we will have to remove the infected eggs. If we do not do that, the entire clutch is gone. Okay, so this is what happens on day 31, where based on the molds, we can say that yes, only one made it to the adult. So that's 1 out of 15 eggs, and that's a bad number. So this is the danger of having them in contact with our fingers. We can wear gloves, no problem with gloves, but try not to touch them. So day 21, these are very beautiful. This you can handle with your hands, no issues. The moment they start taking in paravetillin fluid and expanding in size, they are safe for touch. Before that, no. So sometimes we get nests like this. So we get curious, we stand to put it on our finger, and then we start taking pictures just to see how they look inside. Okay, but that method best is to be done in the laboratory. So given the chance of a laboratory, we will use the image in the middle, the egg incubation. We will use petri dishes. We will put the embryos over there and put them under the dissecting microscope. And then for the spawning cycle, this is what we will do usually. We will look for the horseshoe crab adult, and then depending on the time of the day, we will observe them. If we are not going during a high tide, we will go during the low tide and search for nests. And then juveniles is where my students were successful in Pandas, in Balo. They were successful at getting it. But throughout my experience, maybe the working hours were different. So I did not have a chance to encounter them in Malaysia. But in Thailand, if we go to Pechaburi or we go to Bay of Songkhla, 120% you will get something. Okay? So this is what they did. They learned about after the first embryonic mold, what happens to the horseshoe crab because it gets hungry. Then they started testing. What does the horseshoe crab eat? So they started giving it egg custard, rotifers, what do you call this, like coffee pots. So they started giving them and seeing their reaction. Sometimes they will predate, sometimes no, they're quite picky. Okay? So over time, they have come up with this diagram just to guide young researchers, the do and the don'ts. So what are the safety measures to take depending on the experiment sets that we are carrying out, and what are the best practices? Now, this is my personal diagram from the master's training right until PhD, and the same diagram is used to teach the next set of scholars. So when we go to the field, among the considerations that we are looking at is the inclination. A shore that is very inclined is very unlikely to have horseshoe crabs coming there and nesting. A good or gradual inclination is what we want. But when we go there, we have to make sure the beach is soft. So when we step on it, it leaves a footprint. Those kind of beaches are very favorable for horseshoe crab spawning. So aside from that set of keys, we also need to teach our new set of scholars what is going on. When the horseshoe crab eggs are buried, they go through two cycles. One is known as the inundation, where the tide rises, and then there's another one known as tide fall, where we call it as drying. So these are the two cycles that are very important to the horseshoe crab eggs because it provides them with osmotic shock, periods having high salinity, and then periods of dryness. So they need this kind of shocks. And recently they discovered that thermal shocks, that means heat shocks from the daytime, play a big role. Because thermal shocks provide momentary protein excitations so that it stimulates them to develop. And with that, they discovered leaving the eggs in the field, depending on the position on the high tide. The viability of the eggs are very high when it's higher at the tide than it is at the lower section of the beach. So this gave me an idea of in year 19, how do we investigate the beach? Are we going there just merely to collect sediment samples or looking at the texture, or are we looking at shifts in the sediment shore from fine grains to coarse grains? Because these shifts are the ones that will determine whether that area is suitable for horseshoe crabs to come. So this is the guide that we are using, and it's very straightforward and easy to understand, but it will need more polishing over time. And then this is what happens sometimes when we go to the nest. We are getting larvae that has hatched and ready to go out. So it means that during the lunar tide, the highest tide will come, and these guys will slowly crawl out, like in this image, the last image over here. So they will crawl out, and then they'll follow the tide to the sea. So this correlation in their behavior provides us a lot of understanding and has improved our knowledge on the horseshoe crab biology. Now, this kind of work is not easy to replicate, but it's a very good article that has been published in 1983. What happens during the insemination process? How do we know whether the horseshoe crab egg has been successfully fertilized? So we will have to look at the periplasm, we will look at the yolk pits, and we'll look at the platelets. So I've replicated this experiment during the master's training, and what I learned was exactly as it was mentioned here. When we do a dissection on microscope, histology dissection, we are able to see the structures under the microscope. But when in contact with heavy metal, that's where the structures start changing shape. So we have the work in when I was carrying out a master's has been published in year 2020, but I used the word embryonic membrane for biological filter is because this structure is very sensitive. If in contact to any form of agents that are unfriendly, it will entirely become like a piece of glass. Nothing can go in. And then the horseshoe crab ceases its development. But what Barnum and Brown did was, when in contact with the male sperm, it allows the sperm to go in, although the sperm is a foreign object in a foreign environment. It allows the sperm to go in. But when we have metals, it is completely blocked. Nothing is allowed to go in. So this unique experiment was able to be replicated at that time, but it's difficult to do it now because of a few conditions. One is the horseshoe crab health itself. So some of the experiments that were carried out in the past, it was carried out very tediously, but only recently published because the work is very difficult to do. And especially like this kind of diagram. We carried out many experiments until we got it right to get the number of days, to get the mode cycles, and to get the development. So this was carried out in America, and this was carried out in Malaysia in our lab. So I would say that if this kind of experiment ought to be replicated, it is possible, but we need a lot of patience because there are risks with the water quality. We cannot just use water from the sea. We cannot just use artificial saline water. We have to filter it. The caking agent, the calcium inside the seawater itself, it will interfere with the development of the horseshoe crab and cause it stress. So I must say that it may look like a very straightforward diagram, but it took time to get this done. Okay. So this is again back to the American. So they did all the mold and how the development will take place and how long does this take place. But what we notice is from the fourth mold to get to 22, it's like between 7 to 11 years, the duration. So that much of patience they had to get this one individual well-equipped, well-fed, well-nourished, able to grow in a controlled environment just to get this diagram. Yeah, it's not easy work. Huh? So what they learned was the male has one mold cycle less than the female. And therefore the female is larger and the male is smaller. Okay. So roughly the entire growth period will take 7 to 11 years. And then this was carried out in Jane Brockman's lab, where she did pieces of experiment based on the bird scavenging behavior, based on the condition of the habitat itself in her area, and piecing out this information slowly so that it is proven to be acceptable. So though it may look like one diagram, but you must observe that it took a very long time to get this right. Yeah. So again, horseshoe crab diet, among the early studies in Malaysia, it was in year 2008, done by Dr. Zaleha Kasim, and then again repeated in 2012. Because the results were less convincing. They ate whatever we gave them, but we do not know whether it's their original diet or not. So what they did was they started dissecting horseshoe crabs at the alimentary tract and removing the contents and finding out what was actually there. And with modern molecular studies, they were able to find DNA and trace the DNA to what organisms they are. But to accurately point out which species, that is very unlikely because what is remaining there is remnants and it is in a clump. So it is, if the DNA is not degraded, they're able to identify. If the DNA is degraded, they're unable to do it. So from there, they learned that horseshoe crab diets consist of various organisms. And they started improving the hatchery formulation because they want to try and see a horseshoe crab culture. There is demand for blue blood, there is demand for horseshoe crab as an alternative protein. So they had to develop this kind of indices using, we call it invasive methods, where the horseshoe crab dies, and then moving on to less invasive methods where we give them what they actually want to eat. So these are the organisms that were selected. And horseshoe crabs have only a very limited range. You cannot get them all around the world. For Asia, it's from India right up to Korea. And then from America, it's only limited to the East Shores. So the number of researchers and along with their universities, most of the work for America is on the east side of America. So these are just the brief information for everyone to see. And now we get to the important aspect of the presentation today, the Quaternary period. In these few sketches, in a brief manner of explanation, I would say we used to do hunting gathering. Okay, then from hunting gathering, we found out which organisms could be habituated, and with that we started cultivating. So agriculture started coming in. We started keeping livestock, and with that came performance. Which village is able to have more food than the next village? And then the leader speaks out, and then came war because they wanted to have more lands. They became greedy. Okay, so the same thing is what we are seeing now. The growth, the increasing human population is creating a lot of challenges for the older and the younger itself. So how do they interact with each other? How do they outperform each other? So that came on to industrialization and the damage to the environment. So this is where we are today in the Quaternary era. And in year 2022, uh, the Atlantic horseshoe crab and the tri-spine horseshoe crab are already listed in IUCN. And this is where it gets worrying, okay? Because we have another two which are available in Malaysia and exclusively for Peninsular Malaysia, but they are not listed in IUCN. They're still known as data deficient, despite the problems that they are enduring. Okay? So the image below is where we are looking at the trade, and this image was taken in Peninsular Malaysia. Okay? 1 ton of horseshoe crabs are going out for trade, but we do not know to where. I was recently contacted by a reporter in America. We tried to get the data from Tritch. It's a website where we can get numbers of exports or numbers of imports, but the numbers are it just doesn't make sense. It's in the millions. Now, where are the crabs coming from? If Malaysia, we are already seeing this. Why aren't the crabs being able to be listed in IUCN. Okay, we have worked on the technical document, but we could not convince IUCN the data is real. Okay, so what we did was we went and learned from the community. That's the only way we know what went wrong with our sensors. So if they want to catch horseshoe crabs, they will use special nets. If they are going out for fish, they will use 4-inch, 4.5-inch. But for horseshoe crab, it is the 8-inch net. So with that, this information was the I would call it an overview of all the brainstorming that we did, trying to find out and see what's going on. How did they make it to the trade route? And this was the solution. The fisheries authorities have no objection if fishermen use larger mesh size because the younger populations of fish are not threatened. But it gets worrying because if they are using this kind of nets for horseshoe crabs, it means that they are exclusively hunting them. Okay, and in a short time, we may lose a huge population. And the meal below was prepared to me by a fisherman, but I declined to consume it. Okay, so I just took the egg and whatsoever, but this is what carcinoscoppers could be made into. Despite people are telling that there's tetrodotoxin, it's poisonous, but the fishermen know how to cook it so that it's not poisonous. Okay? And this kind of knowledge itself offers us an opportunity to find out how can all these horseshoe crabs end up in the dinner table in restaurants being consumed in large scale. Another thing is that because of demand, the prices of the horseshoe crab have gone up. So the supply is there. They are requesting 40 crabs, they get their 40 crabs, but we are not sure where are they getting it from. And the prices keep going up because of the demand. And when we did an interview between the areas, what we learned is that they are only discarding the male crabs. All the female crabs are participated in consumption. So entirely, whether they are with eggs or without eggs, they are taken up because of their larger size and they are sold. The male crabs will end up on the shore like this, left to die. So this is a reality in Malaysia. How do we know it's a male crab? We see the full set of spines. We see the falling organism, we know it's an old male. A male that cannot compete in sperm competition is not able to get a female counterpart. And with that, we categorize it as less fit. And it is these male crabs that are being discarded by fishermen. They don't waste time removing them from the net because the barnacles are sharp. It can cut them, it can damage the net. So what they do is they just discard it entirely. So the next thing that I would like to share is the scale of exploitation that is going on in Malaysia. We are in the Anthropocene era. It's a new era, part of Quaternary. In the Anthropocene era is where we do the maximum damage to our environment. Shore modification is creating a very big impact. It's happening all around Malaysia, but someone has to realize that the impact is irreversible. Okay, so what sort of impact is causing? If you observe in this map, we are actually going through food security issues. And at present, our food security is in a very bad state that we have to create an entire task force to find out ways on repairing the damage that we have done. Okay? The shore damage by ripraps, shore damage by reclamation, constructing artificial structures in the sea. These are all not suitable for tropical areas because we are biodiversity rich. If we go to other areas, it's practical for them because the waters are deep, it's oligotrophic, and the nursery grounds are not impacted. For us, it is impacting the nursery grounds. Without the nursery grounds, how are we going to restock the organisms that we are fishing from the sea? So therefore, This is the result. The thing is that we have to look out now is our leadership. Who is leading decision making? Who is leading our future in Malaysia? If we do not do something, prices of things are going up. The horseshoe crabs will be wiped out entirely. So these are few articles that the ones in green are published by our group, our IUCN Horseshoe Crab group. And then we have a little bit more by a few people who have learned from the experts, and then they write out their experiences. So these are the further readings that are highly recommended. And then these are just how do we make horseshoe crab very interesting. So kabuto or kabuto gani is the name that is given by the Japanese to horseshoe crab. So they have put it on the Pokemon cards, trying to make it look attractive. It's not an attractive animal, but they're trying their best. So these are ways that they are actually telling people we need to protect the animals. We need to protect the horseshoe crab. So if you play Pokemon, when you come to Kabuto Cops or Kabuto, then you know it's about the horseshoe crab. So the image below is the blue blood that is being extracted by horseshoe crab. We do not know whether the results are real or not, but they are saying that all the horseshoe crabs are able to survive the bleeding. So right now, for the entire planet, for the persistence of the organisms itself, we have to start thinking about the future, whether our children, our next generation are able to still experience what we are experiencing today or not. So with this, it's the end. Last slide.
Oh my goodness, what a fascinating sharing. Tell me again, I missed what you said. Why are they extracting this blue liquid?
Oh, the blue liquid is, it's okay, it's like for markers. If we have contamination for medical equipment, like the granula, the catheter. So if we want to sell it outside to make sure that it is sterile, they will use the blue blood. So if it coagulates, then the entire batch is rejected. Yeah.
I see. And so they capture these horseshoe crab to do this.
Yes.
Oh my gosh, this is absolutely, I mean, I'm so glad you're speaking to us, because I'm not just me, but many of us is equally fascinated. So Colette is saying, could you please repeat what you just said about the lifespan? Did you say 19 to 21 years?
Yes.
It's a long time.
Yeah. But only 4 or 5 years of reproduction.
Yeah.
Yeah.
But you see, I mean, generally normal crabs don't last so long, right? The normal ones that we see.
2 years, 3 years, they're
That's
already
right.
Yeah.
But this is very, very long.
Yeah.
And I'm not sure whether I've seen it in restaurants before, but why do people eat it if you say it's poisonous and you need special skill to extract to make it non-poisonous? Why do people like to eat it? Is it tasty or something?
I have no idea whether it's tasty or not, because I myself have not eaten it. But why people like it is because of this word called exotic protein. Yeah, those days people used to eat mantis shrimp, like once in a blue moon, then it became an in thing. Now horseshoe crab has become the in thing.
I see.
Yeah.
What I find absolutely fascinating is you say that they will come out to mate when it's lunar, when the moon, when it's full moon.
Yeah.
How does it work?
Okay.
How do they? I mean, I'm asking a very profound yet very simple question. How do they know it's lunar? Is it the light? Is it the gravity? What is it?
It's the light.
The light.
It's the light. Yeah. So what will happen is that's why they call it the spawning ritual. The females, they will remain in the shallow water. The males are the ones that have a higher home range. So they move around in quite a big area. So what will happen is when they start feeling the water getting cold, they will come back into the shallow water because the tidal cycles are such, coming onto the land, removing the heat from the land, going into the sea, and then bringing in the colder water and cooling down the shore and again going back. So this is how the seas function. So what they will sense is the water getting cooler and when they are in deeper waters. So it means like, okay, the big tide is coming in. So they start moving into the shallow water, and with the lunar light, they are picking up that sense, that cue to say, okay, it is the reproduction time. So picking up the light, they move on, they start looking for a female. They will latch onto one female, move into shallow water, and start searching for areas to spawn. And it coincides with the lunar highest tide.
Fascinating, really. Friends, I'm not going to be the only one asking the questions. Do you have questions? Please either unmute to ask or just key in your questions, friends. I see that you have some colleagues or your team here, Brian.
Yeah.
Very nice. Like Wuti, I recognize that name. So nice. You know, I while waiting for the questions to be asked, I'm just absolutely in awe that we have so many of you who are so keen to find out about our biodiversity, you know what I mean? And to document this so that we all know about it. So grateful that we have scientists like y'all passionate about things like this. Yeah. Yes, Lucita, you have a hand raised.
Hello.
Hi, Lucita.
Hi. Right now I am in China. I'm Indonesian, and thank you so much for the nice talk, Dr. Bryan. And I have a question. I was wondering, you mentioned about the illegal trading, and the number you think is not make any sense, right? Do you think those number are collecting from other countries, like example Indonesia, because we are a neighborhood, right? So like they actually transferred from Indonesia to Malaysia first, or maybe from Thailand to Malaysia. Do you think it can be happen? Thank you.
Thank you for your question, Lucita. Actually, that is what it runs in our mind, whether they are brought in from other places into Malaysia and then going out as a bulk. We are not sure whether that sort of route is available, especially Indonesia, Singapore, Thailand, and it could happen even in the South China Sea. But what we are certain is, based on the website called TRIDGE, TRIDGE, they have the statistics by the country, by the commodity itself. If you are looking at crabs, what sort of crabs, by the species. If horseshoe crab, the quantity or the value based by money, USD. So with that, every country has its own value, but the worrying number is coming from Malaysia. Yeah, it just doesn't make sense because we do not know from where they're getting them. Yeah.
Thank you for your question, Lucita. While waiting for others, please raise your hand or key in your questions, friends. I'm just expressing also my concern about, you say that they are systematically fishing or harvesting this for consumption. We have no idea of the numbers, right? In terms of what is being consumed in Malaysia, what is so we are unaware of the rate of the reduction as well then.
Yeah, we are not aware. The problem now why we cannot get the Casinococcus and the Tachypus gigas listed in the red list is simply because they said there's so many areas that we have not collected the numbers from. These include Bangladesh, Vietnam, Cambodia, um, towards the Andaman. We have nothing there. We do not know who is working on them, who can work on them. So even from Myanmar also, we don't know. We are not getting anything. But recently, fortunately, recently Philippines were getting something through Dorcas Kaiser.
I think just like how tigers, there's a tigers network, there is a, people who work together just to protect a species. Maybe there's a need for that collaboration across the region, right? Because it's such a shame if they've lasted for 250 to 400 million years, for us to consume it in this period, it's unforgivable, isn't it?
That is why we are in the Anthropocene era.
That's right. Is there a big consumption in Malaysia, and is it popular, you think?
Yes, I'm very surprised it's even until Langkawi. Yeah, if we go to Malacca, yes, you get it pretty much everywhere. If you go to Johor, pretty much everywhere. Moving on to Penang, yes, you can get it, but Casino Scopus instead of Tachypus gigas. But Langkawi is what I was shocked to get Tachypus gigas being sold there.
So what you're saying is it's actually not available there, but yet it's transported there for consumption.
Most likely from Pulau Paya, areas that are having mangrove swamps, areas which are having sandy beaches, except the ones that are on the island, because horseshoe crabs, they need areas which do not have tides. The surf is very mild, because with strong currents, they will just go in circular motion. So they need calm seas. So definitely not from the islands. Yeah.
So what is CF and Shimla? They are the friends from Canada. What is the Malaysian government doing to curb this or protect this? Are there any protective measures being or are these what you've just raised as a concern? Is it only among the academicians, or is it being slowly maybe spread to maybe others within the, what do you call that? Is it the fisheries? Do they come under the fisheries department?
It comes under fisheries.
Yeah. Is there concern as well expressed by them? And if so, what kind of things are they doing about this?
Okay. In an overview, fisheries, any form of fin or shellfish is a commodity in Malaysia, unless they are classified as bycatch or trash catch. So if they're classified as bycatch or trash catch, they have already listed a set of fishes or set of shellfish or finfish in Fisheries Act 1985. And along with that, the fishing gears that are permitted. Horseshoe crabs are not listed in anything. That is the problem.
Are there people who are working to include that on the list?
At present, we are trying to engage, because there's a team from UIA, there's also a team from UPM who is working with the fishermen and the fisheries authorities. But I do not know about their progress, because we need to attend conferences in order to know the progress. Yeah.
Really, it would be, like I said, it would be such a shame if this people in this generation or next 2 or 3 generations kill something that's 400 million years old. That would be absolutely ridiculous, really.
Yeah, I agree. Yitmi, that's a shame, because thanks, Brian. Thanks for answering that question. I wanted to ask you, does the fishery department at all consult people like you, or have a department that you can actually express some sort of because this will just fade away into nothing, and the markets will just sell off everything pretty much?
Okay, fisheries department, they work by how do I say, approach and engage. They work by that way. So if the species is not a species of commercial value, we get weighted on meetings, we get weighted on discussions. So until it reaches to their attention, then only something will be happening. Like for example, the development of turtle excluder device, where turtles were getting caught. So you had these NGOs coming and making a lot of noise, and then they decided to have this TED enforced for all the fishing nets. So the same thing goes with the type of fishing gear. We cannot use this fishing gear called Bobo Naga, but illegally it's being sold. It's a long net with multiple components and it catches every single organism within it. So that is banned in Malaysia, but you are still seeing fishermen having that. So back to the question whether we are able to engage DOF or not, it will have to be in their priority list. Then only it will receive attention. Yeah, so at present, the NGOs in Malaysia for horseshoe crab, I would say it's close to zero. There is one that's being developed, but we are still in the developing stages. Yeah.
Thank you. I appreciate it.
You know, then I think how many of us here, we had 28 people, now it's 26. You know, all 28 of us must go out and really advocate this. And if we see in the market, you know, just make sure that we you know, not buy it, go to restaurants, not buy it, and maybe even talk about this, you know, to others. Because if I just feel very sad that if this is something that is left unchecked and there's no one, it's not coming to the appropriate attention, not coming to the attention of the appropriate people, you know, to take action. That's such a shame.
You know, the first day covers post Malaysia release, they released the horseshoe crab being cooked.
Oh no.
And fortunately, we made noise.
Cooked. They actually showed a cooked.
On the stem, on the first day covers.
Yeah.
Yeah.
Oh wow. What a
Says a lot, huh?
yeah. And it's amazing when you showed that the map of the world, and really in such limited areas do we find this, and we are so blessed to have it in our waters, isn't it? So
Yes.
I think it has a lot to do with education, Brian. So how do we educate our people enough? You know, how can we bring awareness? You see, maybe the folks in the kampong that eat the crabs think that it's plentiful. You know, maybe they don't even know that this is perhaps at risk or something. So it's all about education, right?
Yes.
So how do we do that? I don't know.
Actually, to be honest, we are developing something, Wuti and myself, but Wuti wants to focus on a broader scope. So once we get the thing right, then we will narrow down and have it for target species.
Okay. We look forward really to you and your team doing this. And of course, the 25, 26, 28 of us will go around and, you know, help to champion it in our own little ways. Certainly, we are not going to buy this, but that's just one person. We ought to talk about it so that there are more, you know, multiples of our friends will also know about this then. Yeah. Any more questions, friends? It's we have already taken Dr. Bryan 1 hour, more than an hour already. If no more questions, I don't see any hand raised. No. So friends, I just want to let you know, next week we will not have a session because it's Hari Raya week, right? Everyone's going to be very busy celebrating, and so probably will not sign up for any session anyway. So we are also going to take a break. So that's a quick announcement for next week. But you know what, Dr. Bryan, I'm so pleased, so very happy that you're doing this with us. There is a question. Where are you doing the studies with Wuti, Dr. Bryan?
The thing is like this, Wuti has developed a program where he's going to give merits. So for whoever who enroll, they want to move on from one tier to the next tier. So what Wuti and I are working on is to first accredit the software itself. We are trying to pair it with the Malaysian qualification Certifications Board and then see to what extent can we certify. That means how valid can we get the certificate. So like for example, citizen science, we encourage anyone who wants to be a citizen science, we appreciate their knowledge and we certify them. So to make sure that that certificate have a value, we need to buy it with an auditing agency. Only then it carries weight. Otherwise, it's just a piece of paper.
True. Actually, the other thing is whenever we go to the markets or shores, we should take for a rub and then talk about it. That this shouldn't be eaten. You know, this is one way of reaching out in using the social media to say, you know, this is endangered, or the numbers are coming down. We shouldn't be consuming this, not aggressively, but in a educative way. You know, so that others also are aware of this. Yeah. Yes. Really. Thank you so much, Dr. Bryan. I think you just gave us amazing insight into this. Actually, not really even a crab, right? This HSC, it's not even a crab, but it's we call it a crab, and so fascinating that it is 200 to 400 million years old. It comes out during lunar time. Fascinating stuff. I'm not sure how we can thank you enough for you to spend so much time with us and to enlighten us on this absolutely fascinating topic. And not only that, friends, Dr. Bryan is so kind. He has given me a few more contacts that I will reach out to, and I will also get them to speak with us. So I am going to ask all of you friends to unmute and help me thank Dr. Bryan for this wonderful session that we had with him.
Thank you very much.
Thank you.
Thank you, Dr. Bryan.
Thank you, Dr. Bryan.
Thank you, Dr. Bryan.
Thank you, Dr. Bryan.
Thank you.
Thank you.